Welcome to the ISoRED webinar series on community building in radiation epidemiology and dosimetry research. Today we'll have the radiation epidemiology and dosimetry program at the UK Health Security Agency presenting. My name is Kaitlin Kelly-Reif. I'm an epidemiologist at the US National Institute for Occupational Safety and Health, which is part of the Centers for Disease Control and Prevention. I'll be moderating today's webinar. The overall format is we'll have some introductory remarks and then 4 10-minute long presentations with some Q&A after each presentation. You're welcome to write questions in the chat or ask questions at the end of each presentation. I'll be moderating the questions in the chat. So I'll hand it over now to Simon Bouffler, who's the Interim Deputy Director of Radiation Protection Science, to give some introductions and an overview of today's presentation. Simon?
Yeah. Thank you. Thank you very much, Kaitlin. And it's great to have the opportunity to outline some of UKHSA's work in radiation epidemiology and the associated areas. So UKHSA is really quite a new organisation. It was only formed in October of last year, and we're the sort of UK-wide health protection organisation working on infectious diseases as well as chemical, radiation and other environmental hazards. We've had a key role in the COVID response in the UK, and indeed some of our radiation specialists even contributed to that. While being quite a new organisation, we draw on a long legacy really back to the 1970s, with our radiation protection functions really remaining rather similar over that period. Some of you are probably familiar with the original organisation, NRPB, the National Radiological Protection Board, which had a unique focus on radiation protection while we're incorporating this much larger organisation today. We carry out our research really for several purposes. One, to make sure our radiation emergency response functions are up to scratch. Also to make sure our general radiation protection advice is as good and as evidence-based as possible. And it also feeds into the development of our services, which we offer around the country and internationally. And all this also feeds into our international and national advisory work, where we contribute quite substantially to organisations such as UNSCIR and ICRP. And today we've got 4 presentations from colleagues covering different areas. First, Richard will cover our epidemiological work. Tony will then come along to cover some aspects of internal dosimetry. Then John on some developments in external dosimetry. And finally, and maybe it's a little bit different to some of the previous presentations, Christophe will cover work on biomarkers and biodosimetry, all of which I think are relevant for the radiation epidemiology. So I'll hand back to Kaitlin to take us through.
Great, thanks. So first up, we'll have Richard Haylock. He's going to give an update on the UK Radiation Worker Epidemiology Program. He's the group leader for radiation epidemiology. Go ahead.
Thank you very much, and welcome to everybody. Yes, so the UKHSA Radiation Worker Epidemiology research is based pretty much around the UK National Registry for Radiation Workers, which is maintained and run by my group. This is the NRRW was set up in 1976 for radiation workers whose employers agreed to participate, and it's now grown to around 277,000 workers. So the largest employers that contribute are our Ministry of Defence, so a lot of these are like shipyard workers. We also have workers from British Nuclear Fuels Limited, although that company doesn't actually exist anymore. Another big contributor is the UK Atomic Energy Authority. We have a group that I've put together and called the Power Station, so these are the nuclear power stations in the UK. And finally, the Atomic Weapons Establishment. So those are the sort of major groups that we have. Other groups are smaller, and the other group is sort of all the rest. So we collect employment information and personal data about all these people to enable us to follow up the workers in the future. We also collect individual recorded external dose data and fact of internal monitoring, but we don't actually have internal exposure measurements. Currently, we don't have any information on smoking or other factors affecting health, but we hope to change that in the future, though that's a bit of an aspiration at the moment. Our follow-up data consists of mortality data from 1955 and cancer incidence data from 1971, when that first became available nationally in the UK. There have been 4 major analyses of the cohort. However, we call the latest analysis NRRW 3.5, as it was based upon the same workers as the previous NRRW 3, but with an additional 10 years of follow-up. It examined 167,000 workers, of whom around 10% were women. The mean follow-up was just over 32 years, which isn't quite as good as the lifespan study, but still pretty good for this sort of study. Cumulative lifetime doses are small with a highly skewed distribution. About 2/3 of our workers having doses less than 10 millisieverts, and only about 6% having doses of above 100 millisieverts. This figure illustrates the analysis of malignant neoplasms excluding leukemia based upon the NRRW 3.5 dataset, and shows a statistically significant linear excess relative risk with dose. The categorical point estimates do suggest a certain amount of leveling off of the excess risk at higher doses, but overall, the linear model is in reasonably good agreement with a similar model fitted to a subset of the lifespan study data, that's a lifespan study cohort, of men exposed between 20 and 60 years of age, with dose limited to 2 gray. So fairly good. The analysis of cancer in the NRRW 3.5 dataset did not just look at large groupings, but also examined a whole range of individual cancer types. This figure presents the results for the for the 20 cancer sites with the most cases. Compared to the overall groupings, you can see the numbers of deaths for the individual sites were much lower, and therefore the power to detect excess risk was also lower. Ooh, that's one too many. However, we did see good evidence of raised risk for several individual cancer types, e.g. rectum and bladder, as I've highlighted here. In NRRW3, evidence of excess risk for non-cancer diseases, such as heart disease, was limited, but the extra follow-up in NRRW 3.5 provided many more non-cancer deaths, and we now see the first good evidence of excess risk. In 2019, my colleague Wei Zhang published a paper looking at risks of heart disease in this NRRW 3.5 dataset. Wei found good evidence of excess risk with radiation dose. Over the whole dose range, there was good evidence for non-linearity in the dose response, which was clearly driven by the lower risk at these higher doses here. Whether this is really evidence for a nonlinear dose response or the result of other factors such as the healthy worker effect will need to be studied in more detail in the future. So we are still working with the NRRW 3.5 dataset and have 2 papers published so far this year. Nazos Hunter from our group has just published a paper on lymphoma and multiple myeloma incidence and found evidence of excess risks for non-Hodgkin's lymphoma and multiple myeloma, but not for Hodgkin's lymphoma. While Catherine Hinksman has examined cerebrovascular disease and found excess risk with non-linearity similar to that for heart disease found by Wei Zhang. I think that this paper is published is available ahead of publication on the journal's website, but the actual paper should be published shortly. Our group is working on several further analyses of the dataset at the moment, including an analysis of nonlinear dose-response relationships for solid cancers using parametric rather than stratified baseline models as we've mainly used in the past. We are also investigating respiratory diseases and participating in the InWorks 2 project, which is pooling worker data sets from France, the UK and the USA. And the UK contribution is based on the NRRW 3.5 dataset. Turning now to developments with the database itself. While the UK HSA owns and manages the NRRW, we also manage 3 further long-standing radiation worker cohorts, the British Nuclear Fuels cohort and the UK Atomic Energy Authority cohort, that are both owned by our Nuclear Decommissioning Authority, and the UK Weapons Establishment cohort, which is eventually owned by our Ministry of Defence. While most of the radiation workers in these other cohorts are also in NRRW, these cohorts do hold individual internal radiation exposure measurements, and they have large groups of non-radiation worker employees that can act as control groups. The NRRW currently has no internal exposure data, nor a non-radiation worker comparison group. We now have the agreement with the owners of these cohorts to merge them into NRRW, and we expect this to happen in the next year. As a result, NRRW will gain internal exposure data for about 30,000 workers and a non-radiation worker control group of about 70,000 people. So we think this is going to be very useful indeed. Finally, to the future analyses in the NRRW, and that is, of course, our NRRW 4. We plan to start this in 18 months to 2 years' time, and expect a large increase in the number of workers we will include, up to maybe 250,000. We will also have another 10 years of follow-up and expect about 65,000 deaths. The analysis will examine the effects of both external and internal exposures on risk of both cancer and non-cancer diseases. So we expect to work with this cohort, this data set we extract from the cohort for a number of years, and hopefully published a good many papers based upon this data set. Finally, I just need to say that the running of the NRRW is very much a team effort, and I'd like to thank the hard work and dedication of all my group that go into supporting it and keeping it running. We realise we're only custodians along the way, and there will be people after us that will take this cohort into the future, we hope, and maybe have it running for another 50 years. And that's everything. So
great.
Thank you very much.
Thank you, Richard. That was a great talk. Let's see if we have a question in the chat from Elizabeth Cardis. She says, in terms of international exposure, what will you have? Just the fact of having been monitored? Individual dose calculations would be difficult in the cohort.
Internal, not international.
Oh, I'm sorry, internal.
Sorry, yes. No, we will take, we will have individual level internal exposure measurements, and we do expect to be able to calculate, well, we will not be calculating individual doses. That will be the responsibility of Tony Riddell, who's going to talk to you shortly. So yes, we do expect to have individual internal doses.
But individual measurement means levels from urine assays or fecal assays and things like that.
Yes.
So you'll have all of that data in the NRRW.
Yes.
Okay. That's great.
Any other questions? I have a question, Richard. I was impressed with the variety of different types of industries that comprise the cohort. I was wondering if there was any information on non-radiation exposures, such as chemical exposures. You know, in the US, we have some unique chemical exposures as part of the uranium enrichment process and nuclear working processes. Have you looked at those, and have you looked at the interactions between radiation and those other exposures?
We haven't particularly. We do have some information on the cohort about people's prior exposures to industrial chemicals, but it's not particularly complete across the cohort. So no, I mean, for example, we'd like to know more about things like asbestos exposure, but no, we have it for some. We hope in the future to collect more of that, but at the moment, no, I'm afraid not.
Thanks.
Richard, have you got any plans to link with other NHS data sets, and in particular to get things like hospital episode statistics and other such, which obviously be a huge amount of work, but it would enable you to get perhaps a better handle than just mortality on some of these non-cancer endpoints? Indeed,
we would love to be able to do that. The ethical route to doing that, the ethical approvals route, is quite challenging to say the least, particularly as now all this data is controlled by NHS Digital. It's moved out of Public Health England as we were, and we didn't carry it over into UK HSA, but it does exist, and yes, we would love to do it, but we're working on our current next step is integrating these other cohorts into NRRW, and then moving on to looking at other areas. Yes, the challenge of getting ethical permission to get that data in is not insignificant, but yes, I'd love to do it.
Thanks, Richard. I think we don't have any other questions. Let's move on to Tony Riddell, who's going to talk about internal dosimetry. And if we have more time at the end, we can have a broader Q&A. I'll hand it over to you, Tony.
Thanks. Hello, my name's Tony Riddell. I'm interim head of the Radiation Hazards and Emergencies Department, and I'm also the group leader of the internal dosimetry group within that department. And I'm here to talk to you about internal dosimetry work related to epidemiological research. Okay. As Richard mentioned, UK HSA manages several worker cohorts, including the former BNFL sites worker cohort. And this includes major sites such as the Springfields nuclear fuel production facility, the Sellafield reprocessing plant, and the Capenhurst fuel enrichment plant. And as you also mentioned, these are significant because they include large numbers of workers with potential internal exposures. So there are over 30,000 workers with potential internal exposures, mainly from uranium and/or plutonium, but also there are reasonable sized cohort, 2,000 tritium workers, and some of the radionuclides such as fission products, etc. For these workers, we have over 1.5 million urinalysis results at the moment. We're currently receiving an update on that, so that number is likely to go up. There's a separate dosimetry database for epidemiological research for the BNFL cohort, with doses calculated from these urinalysis results, and it's believed to be the largest in the world with organ tissue specific internal doses. So you might say, well, if you've already got this database, what do you do? So the ongoing drivers for what we do are to improve dose estimates. Internal dosimetry is far from a complete science. New methodology arrives all the time, and in the lifetime of this cohort, which extends past UK HSA's involvement to the time when I worked for British Nuclear Fuels, we've gone through several revisions of ICRP methodology, 30, 60, 66, 78, and we're currently working on the 130 series, which is the occupational intakes of radionuclides series of publications. We've also used non-ICRP methodology when we thought that that was better at the time, including the Jones excretion function for plutonium and the latest Leggett plutonium model before it was adopted by the ICRP. We've also used model parameters and assumptions that have changed over time as our knowledge of exposures and the materials people were exposed to has improved. We've also got a lot of issues, although we've got a lot of data. It's not perfect. We're looking at the analytical limits of detection quite a lot of the time for a lot of these measurements. So we've got quite a lot of sensor data, particularly in more recent times, as internal exposures have got smaller. We have periods when we know there is no urine data, or the data that we have is unsuitable for accurate dose reconstruction, or potentially unreliable. And as I've just mentioned, we also from time to time receive updates. Some of these cohorts are still live, although British Nuclear Fields no longer exists. Sellafield Sites does, and this covers many of the workers who were formerly with British Nuclear Fields. Something else which has come to the fore over the last decade or so is estimating uncertainties on these dose estimates. And this is a far from trivial topic, and has taken quite a lot of time and thought, and again, is something that we're still working on. The other thing that tends to happen is that it's important when you're doing pooled analyses or joint analyses of multiple cohorts that you used harmonized dosimetry for across all the cohorts involved in those studies. So we may have to recalculate internal doses to make sure that they comply with the dose protocol produced for an individual study. And some of the major studies we've been involved with over the years include Alpha Risk, SOL, SOLO, and IPOR, which is an international pooled uranium worker study, which I'll talk about a bit more in a moment. To back up our work and updating the doses in the database and dosimetry data, our research involves looking at improved and updated methodology. Some of the members of the internal dosimetry group contribute to ICRP work. We have links to the US transuranium and uranium registries, and we've done things like look at plutonium binding in the lung along with them. Because internal dosimetry requires quite extensive calculations and data fitting, we have been heavily involved in developing internal dose assessment software over the years. Some well-known examples of this are IMBA and our recently released Taurus software. And I'll talk a bit more about Taurus in a minute. As we're looking at very large cohorts of individuals, updating or replacing doses is obviously very time consuming and would not be possible on an individual basis for this cohort or sub-cohorts of it. So we've been heavily involved in developing automated internal dose assessment processes so that we can fairly rapidly update or replace the doses in the database. Because internal dose reconstruction relies not only on the monitoring data, urinalysis mainly for individuals, but also about your knowledge of how they may have been exposed and what to. We've also been involved in looking at historic information on working practices and the monitoring that was done to see if it is suitable or how it may be modified to produce good internal dose estimates. As I've mentioned, sometimes for some parts of the cohort, monitoring information is lacking or sparse. So we've also been involved in trialing job exposure matrix methods for dose reconstruction, and we did this for early urinalysis. For early plutonium workers at Sellafield, using a semi-quantitative method where we used available urinalysis data as surrogates for that where it was lacking. So the latest developments are, as I mentioned, that we're currently in the process of moving over to the latest ICRP recommended methodology for worker exposures, which is the ICRP 130 series of publications, which is nearly now complete. Part 5 is imminent. And this is known as the occupational intakes of radionuclides methodology. This is actually implemented from a practical perspective through our new Taurus software. A base version of this was released just over a year ago, but we are actively developing Taurus to make it more suitable for more complex assessments, and particularly in this context for internal dose reconstruction for epidemiological purposes. And some specific developments for that include what's known as a batch mode, which I'll talk a little bit more about in a moment. And because Taurus was in the first instance developed for protection, operational protection purposes, it largely produces, primarily produces effective doses which are not suitable for epidemiological analysis. So we've been working on production of annual absorbed doses in grey to organs and tissues as an output from Taurus, and this can be fed into the database. As I say, we've got a long history of working with automated assessment software to assist this process. We've been updating that so that we can now use Taurus as a calculation engine rather than previously we used IMBET or purpose-made dose reconstruction calculation modules. And this utilizes Taurus in its batch mode. And also, as previously mentioned, our latest epidemiological study is the IPAWS study. Okay, then, well, hopefully, if I can change my share here. I should be able to show you Taurus. Is this showing okay?
I can see it.
Yeah. So as I say, this is our latest Taurus software. Some people may or may not be familiar with it. Obviously, there's not a lot of time here to go through it. But to give you some idea of how it works, you select a radionuclide here. U234. You can input an exposure pattern here, which can be by different routes, and it can be acute or chronic, to different forms of material, chemical forms of materials. And if you're reconstructing doses, you can enter bioassay measurements, be the urine, etc., etc. And you can go away and do a calculation. Because time is limited, what I'm going to do is to load up an assessment already. So this is a pre-prepared assessment for a uranium worker. Something that's new in Taurus for internal dose reconstruction for epidemiology is that we now have nuclide mixtures. So this is a mixture of uranium isotopes which corresponds to natural uranium. So it's a mixture of U-234, U-235, and U-238. This is saying that we have an inhalation exposure to a 5 micron AMAD aerosol, which is a mixture of chemical forms which approximates to a third of each ICRP default type of FMNS, fast, medium, and slow, which was something we developed during the Alpha Risk project as being a good approximation when the actual true chemical form of the uranium compound was unknown. This is saying this individual has got 2 chronic exposures with start and end dates. They've got 344 urinalysis results, which have a time collection period, which is usually normalized to 1 day. The activity in that sample and whether the sample is less than limited detection or positive. There's also an uncertainty on that estimate, which in this case is a log normal distribution with a GSD of 1.6. So if I then click on this button over here, unfortunately, somewhat in the way. This is typical with these things. I tested this this assessment a couple of times, and there we go. It ran a lot quicker than this. Okay, you can see here now that for both these chronic periods, that there is now an intake estimate within that period. And as I said, at this top level here, there's an effective dose resulting from those. But what is different here now is that because this is a development version of Taurus, we actually have absorbed dose estimates for this individual, and this gives absorbed doses to individual tissues or whole organs in specific years across the period specified, and is broken down into low LET radiation, X, beta and gamma radiation, and the high LET, which is alpha particles, and neutrons from spontaneous fission here as well. Okay. So as I say, that speeds up the actual process of calculation for an individual. But to really speed up the overall process, if I get rid of that. Here we have a DOS box, and you can actually run Taurus just as a calculation engine. So this command line here is calling Taurus with the name of a file which has got the details of the proposed exposure regimes, bioassay data, and the sort of assessment you want to do within it. And if you just enter that, it goes away, picks up that file, doesn't start the GUI, and returns back to the prompt, but it's produced an output file. From which the intake for the individual in years can be extracted. This is collated into another file. This is just one individual here, the one we've just seen.
Hi, Tony, this is Kaitlin. I think we have to move on to the next speaker fairly soon. If you want to.
Okay, yeah, just
Is
about
now-
to wrap it up. Yeah,
Okay, great. Thanks.
yeah, just about to wrap up. Okay, so and these doses are picked out as well. So okay, so just to finish off, I promised to speak about IPOR. This is an international pooled analysis of uranium workers. The UK arm of that is the Springfields cohort, where there are 9,400 workers with over 11,000 employment episodes and 895,000 urinalysis results. We're just this week finished recalculating the doses for them using ICRP-IR methodology, assuming inhalation intake of a natural uranium isotopic mixture of a default of a third of each solubility class. This has yielded annual intake and organ tissue absorbed dose estimates for the period 1949 to 2020 inclusive. And it says, as it says here, there are 16.7 million doses in that file. In fact, there are double that number because there's a high and low LET. Obviously, not all of those will be used within the IPOR study. One other thing to mention before I finish is that this automation process of the assessment opens up the possibility of repeating assessments with different parameter estimates on an automated basis, and this allows you to produce uncertainty estimates on the doses, and this is something we'll be picking up again. Okay, so any questions?
Thanks, Tony. I see in the chat that there's a question from Elizabeth. In the interest of time, we're going to move on. And if you could answer that question in the chat while the next presentation is going, that would be great.
Okay.
We're going to move on to Jonathan Eakins, who's going to talk about developments in external dosimetry.
Hello, everybody. Hopefully you can all hear me.
We can hear you. Thanks.
Going to share. It's doing that. Okay, so hopefully everybody can hear me and see my slideshow. Hi there, so my name is John Eakins. I'm going to be talking about developments in external dosimetry today. So obviously that's a very wide field, and in fact all I'm going to be doing is just giving a little whistle-top tour of two particular aspects of it. So the first is actually of global interest within the field. It's the new ICR report that looks at redefining operational quantities. So that's the big broad brush aspect. And the second, I'm looking at the Podium project, which is a project that involved our group quite a lot, looking at real-time dosimetry. So this is much more focused on the UK HSA side of it. But actually, I'll hopefully show at the end that there is some overlap between these two approaches. Okay, so looking just first of all at the ICR review report, I'll start actually with giving just a little bit of background on where we are. Because of course, you'd all be aware that the current gold standard for assessing radiological risk to individuals are the detection quantities as defined by the ICRP. And of course, as you know, these are defined in terms of radiation tissue weighting doses following biological exposures. So they're based on exposures of people. So I'm using here an example, effective dose. Effective dose was calculated by computer modeling, Monte Carlo modeling of an anthropomorphic phantom. So to see in the picture here, we have a very realistic version of a real human being, which within the computer model gets exposed to the radiation, and the computer calculates the doses to the various organs within it,
which it then sums by radiation weighting factor and tissue weighting factors, and average both sexes, and comes up with the effective dose for the person, so it's a biological estimate of risk. But of course, this comes with a penalty in that it can't actually be measured short of cutting the person up and seeing what doses their various organs got. We can't actually work out what the effective dose is. And this then leads to a problem in designing dosimeters and instruments that are used to assess risk of people. Okay, so enter here the ICRU, and their solution is to define operational quantities, which can be measured and can act as good surrogates in principle for those protection quantities. And then once we have these quantities, these operational quantities, we can calibrate dosimeters and instruments in terms of them. And there are a bunch of these different operational quantities. I'm familiar with of HPD, so often HPP10 is used, and H star D and H prime D for area monitoring. And the idea is that they're defined using simple geometric phantoms that represent people. So it's been found that if we do away with the realistic voxel anthropomorphic phantom, we instead replace it with a person-sized lump of homogeneous tissue, if we were to tally, if we were to record the dose at 10 millimeters depth in that surrogate phantom, we get something that's actually pretty close to effective dose in many cases. And in fact, we can go a step further than that. We can get rid of that person-shaped phantom and actually just use a geometric phantom. In this case, a large slab phantom that can calculate the dose at 10 millimeter depth in that. And that gives us HP10. We find that that's pretty close to effective dose in many cases. And this is not useful because this can be measured. But it does have problems. Now, of course, for a measurement and for a dose quantity, we don't want to underestimate the risk of the person, because of course that could end up being a hazard to the individual. And we also don't want to overestimate the risk. So we don't want to add in unnecessary precautions with their costs in our radiation protection. And it's been found over the last, I think that's over 20 years since ICU proposed those simple surrogates, that these two requirements are not met, or not met very well, for all particle species at all energies, angles of incidence, and for each dose quantity. And so in response, a couple of years ago, ICRU proposed redefining these operational dose quantities in terms of protection quantities. So in other words, they're using the operational dose quantities, they define the operational quantities in terms of organ and effective doses. So for example, HP 0.07, which used to be, or currently still is, the operational quantity of skin, is being proposed to be defined in terms of the actual dose to skin. And the HP10 and H star 10 have been defined in terms of effective dose. And conservatism is being enforced, because remember, for operational protection, what we want is conservative quantities. We want to give a conservative estimate of the dose the person's getting, so we don't underestimate their risk. So HP10, for example, is proposed to be replaced by HP, which for a given angle omega here is going to be defined as the maximum of the effective dose from angle plus omega and angle minus omega. So for example, HP at 90 degrees is going to be whichever is the greater of the effective dose for left lateral and the effective dose for right lateral. So whichever of those is bigger, that's what HP will be at that angle. And similarly, H star 10 is going to be replaced with H star, which is the maximum effective dose for a whole range of different exposures. So again, the maximum conservative dose quantity. So the current status is these proposals were given in draft in November 2017 for consultation to the public community. And I think it's fair to say that there were a mix of responses, which, to give my summary here, that's my opinion, I would say the majority of those responses were in favour in principle, and the majority were opposed in practice. Nevertheless, ICRU went ahead with publication with, as a joint publication with ICRP, as report 95, operational quantities for external radiation exposure. And this came out a year or so ago. And although coming out a year ago, it's unlikely to be in national legislation for at least another 10 years or so, based on how long these things tend to get. So it gives plenty of time for the community to react. And in fact, the UK HSC is currently leading with university institutes from across the world, actually, a URED-sponsored project trying to evaluate the impact of these new dose quantities. And it's hopefully this report will come out within the next few months. It's in its final draft stage now. UK HSA has already published a few papers on the impact of some dosimetry systems. Other teams around the world, other teams around Europe have also published some similar papers looking at the impacts of all these different systems of dosimetry. And they will be impacts because by definition, if we have a dosimeter instrument that's currently designed to be optimized in terms of the current quantities, if we change those quantities, by definition, it's likely that those responses will suddenly weaken. It's likely that we will have negative impacts rather than positive or mixed ones, so they may still happen. So I'll give an example here of the UK HSA personal TLD. This plot on the left is its current response, which is pretty flat across a wide energy range to HP10. But if we to change the dose quantity to HP, suddenly we get this enormous over-response at low energies. And these low energies are really quite important. It's the sweet spot really for X-rays that are used in hospital, for example. So we'd find that a good monitoring system would suddenly start to perform quite poorly. And this means that dosimeters and instruments will all need to be redesigned and recalibrated and tested. And this will, of course, cost time and money. And as I said, I focus also on the recalibration and retesting. This then requires an international response. So we have ISO, IEC, and IEA standards that govern the calibration and testing of instruments and dosimeters, and they're all going to need to be revised. And this is also, of course, costly and time-consuming. The secondary standards based them also no longer be appropriate, and they will need to be developed. We need to be transfer instruments to port things across. I'm giving just a very small snapshot of the different impacts here, actually. But one particular of interest, perhaps, to the current audience is that there will be an indirect change to dose records and registers. So it's likely that the old and new dosimeters will have to be used in parallel. We can't make a change simply overnight. So we'll have two conflicting systems of dosimetry being used essentially at the same time. We'll find that doses received by workers will change. So because we're changing the dose quantities, even though an exposure to a worker may be identical from one day to the next, suddenly we'll find they get a different dose because we've changed the quantity with which we're measuring the dose. And this also will affect analysis of historical versus future doses, because we're changing the dose quantities. A dose from one day to the next will change, or from one decade to the next will change. And of course, this could be quite a headache for epidemiologists who study trends of doses over a period of years and decades. Okay, so I'll come to the second thing I was going to talk about, the podium project, looking at real-time dosimetry. Hopefully, if there's one thing I've convinced people from the first part of the presentation, is that there are actually lots of problems associated with using dosimeters. And so this led to the research question, well, could we use computational methods instead to calculate doses in real time? And this would have a whole bunch of advantages. So, for example, we wouldn't necessarily need physical dosimeters, so we needn't worry about lost dosimeters or workers not wearing them. We wouldn't even need operational quantities, which resolves all the issues we've just been talking about. We can go straight to effective dose or doses to organs and so on. And it can be done in real time, so this gives faster feedback to work. And this then led to the podium proposal. The podium was an EU-funded proof of concept project, so it's really just in its infancy, it's just its early stages, looking at improving occupational dosimetry via computer simulations, essentially. So in other words, no physical dosimeters. And the idea was sort of fairly simple and in two parts. So the first part, and I use this diagram at the bottom, was to use Monte Carlo computer simulations to determine a dose rate map. And that dose rate is going to be in terms of effective dose or individual organs. So we'll build a realistic Monte Carlo model of the exposure geometry, so the laboratory, say, or the facility, and run it to calculate the dose rate, effective dose rate, as a function of location. On top of that, we use tracking cameras and software to monitor the motion of individuals in that field. And this then gives a time-dependent journey of that individual within that field. And by combining those two technologies together, we can come up with real-time assessments of dose as the individual moves around that radiation dose map. Okay, I said it's just a proof of concept, but it was validated within Podium for two situations. So first, these were neutron workplaces, and the second was interventional radiology. And given time, I'm only going to talk about the neutron workplace, and in fact, I'm only just going to give a few slides. But I give on the photograph here on the left, this is our neutron facility at UKHSA. It contains an ambi field, so an amberous and beryllium field is inside this yellow here. This yellow is a lead shield, so it's inside here somewhere, which we then water moderate to come up with a very interesting looking experimental field. Okay, so we then build a Monte Carlo model bit, so that's the figure on the right here, which we can then run with our Monte Carlo to come up with interesting things such as the fluence spectrum, so the spectrum of the field at various locations is a function of angle. And in fact, if we do this at a whole bunch of locations within the facility, we're able to come up with an effective dose rate map. So I show here, and obviously the numbers themselves aren't so clear, but we have this family of dose rate maps depending on the orientation of the individual, because of course effective doses is an orientation dependent quantity. So if the individual is facing the source, they're likely to receive a higher dose, for example, than if they're facing away from the source, for example. So it changes the function of orientation and location. Okay, so that's the first component. The second then is the people tracking. So we set up a camera in the laboratory, so we can see here on the left on its tripod, and this is what it's seeing. This is its field of view, and these are the water moderators that I showed in the previous slide. And then we can track individuals as they walk around this field. So we had somebody walking around, and the camera would track their motion every second. So every second it would say where that person is located and also their direction. So you can see it superimposed across the chest, the direction which the shoulders are facing. And generally the direction which the shoulders are facing is the direction the person's facing. So we see their orientation. And of course, by combining this with the previous dose rate map, we can come up with some dose data for the individual as they moved around. And these are the kind of results we found. We found that these are the spots on the top left. These are the spots where the person was at each second for this test. And of course, with each of these data points, we also know the orientation which they were facing. And so then combining that with the dose rate maps, and of course, this is just one of the 8, depending on this is the person facing, in this case, towards the left. By combining these, we were able to find the effective dose rate at each second. So as the person moved around, their effective dose went up and then shut down a bit, presumably as they walked away and turned away, and then they came back close to the source and faced it, and so on. And by integrating this, we can come up with their accumulated dose as a function of time. And again, this is quite interesting. We found in this case an effective dose. So again, we're monitoring effective doses here, not normal occupation doses. And we found about a microsievert in a minute. So 60 microsieverts an hour average, which is a very small dose. This is much smaller than conventional dosimeters would be able to measure. And of course, it's to a dose quantity that's far more relevant to actually protecting people. So that brings me then just to summary. I'll try and bring these two projects together a little bit.
Hi, Jonathan. This is Kaitlin. I'm sorry to interrupt. We're at 15 minutes now.
Okay,
so
sorry.
Well,
this is my final slide, which I'll let people then read in that case. I'll just say the idea then is that focus may shift more towards protection quantities going forward. This is supported by the ISoRED recommendations. So personal dose needs to be able to provide accurate estimates of effective dose more than they do. But there'll be significant cost burden to this, and there'll be a discontinuity with past data. And similarly, with online dosimetry, it'll also give direct estimates of V. This is something that's likely to be, although it's a proof of concept, this is likely to be developed significantly in the future through the ISoRED strategic research agenda. And it also, by doing dose rates and by looking at effective dose, it will also produce a discontinuity with past data. So again, in summary, and for the interest of the people in the audience here, these changed doses and estimates will affect epidemiological analysis when we look at past and future data. So thank you, and apologies if I overrun a little, but I should stop there.
Thanks, Jonathan. You have a great talk. You have a bunch of questions in the chat, I think several from Elizabeth Cardis, one from Charles Adams, and then I think, Tony, you have another question from Tim Talby. So if you guys could answer those questions in the chat as we move forward, that'd be great. We're going to move on to Christophe Badie. He's going to be talking about developments in biological dosimetry. I think we're going to go a little bit over time, which I think we kind of planned on doing. So in case people have to leave at the hour, I just want to make 2 quick announcements. One is that after this seminar, trainees and early career researchers are invited to attend an extended discussion. Lydia Wilson is going to post that information in the chat. And then this is the last of the ISoRED webinar series for this particular series. There's going to be a new webinar series in April focusing on soon-to-be published papers. And I think Aranya has posted that information in the chat, but maybe she can put that in as well. So I'll hand it over to you, Christophe. I'm sorry we're a little short on time, but you can, I think, go over a little bit, and then if people want to stick around for questions, we can do that. Thanks. Okay, thank you. Can you hear me well and see my screen? Yes. Okay, yes, well, I hope most of you will stay for my talk. I'll try to be relatively quick. Yeah, good morning and afternoon to all of you. What I would like to do briefly, of course, is to give you an overview of the biological dosimetry work project and new development performed in the radiation effect department. So I think because of the time, I'm not going to describe all the different approaches for dosimetry. You obviously know about the physical dosimetry. There's also the clinical dosimetry, and that's mostly for high doses where you have a clinical sign which are clear and indicate a high dose exposure. And then we've got the biological dosimetry, and that's what I'm going to focus on. The more classic biological dosimetry is the cytogenetic part, and then move on to the gene expression one, which is where I'm more involved. So perhaps I wanted to, having in mind, perhaps you know, biology is not the background of all of you, is to give you an idea of what we mean by chromosomal aberration, which we assess, and the number depend on the dose to which individual might have been or have been exposed. So here you've got a premature condensed chromosome. Those you don't need to culture, you can look at them straight away. And here you can see, you count all of these here, the small one, the spaghetti's one. And if you do that, you can really look carefully at what happened over time. This is the time in minutes after an exposure, and the cell is repairing. These are fibroblasts, human fibroblasts, so they're repairing, and that's a cell line which has trouble to repair. So they are repaired, but sometimes you've got unrepaired, and unrepaired are these micronuclei here you can see. Sorry, these micronuclei can be counted and would increase with the dose. And you also have the misrepaired one, and that would be, for example, the dicentric here you can see, and you can again count them. And you can also have a look by painting this chromosome at the translocation. And that's also a good indication of the dose. So in terms of biological retrospective blood dosimetry, what has to be done is to identify, obviously, the individual potentially exposed, take a blood sample. For cytogenetic, you need to prepare and have the cells in culture, the lymphocytes, and then you measure the radiation-induced signal. You compare these test results with a calibration curve for those response which you have generated in your lab, and then you can inform on the dose and the clinical treatment decision help. So I will obviously not go into details more, but we at UKHSA in our department, we provide a specialist chromosome dosimetry service, and that's led by my colleague Liz Ansbury. And we have a lot of collaboration worldwide and involved in the BRANET, which is the International Atomic Energy Agency Response Assistant Network. BioDoseNet is from the World Health Organization. And at the European level, we've got RENEB, and this European network is pretty good as organizing annual training exercise, and also almost every year or every 2 years we've got this inter-assays comparison, which help everybody to
to improve their protocol and their accuracy in providing those estimates. So now, I think it's about 15 years ago, I think I was asked if we could potentially look at other biomarkers. And that was, the idea was in case of large scale exposure to ionizing radiation. Because in such large scale, where you might have 10 of 1,000 people might might have been potentially exposed, then you would need early diagnosis. And that's where the limitation perhaps is with the classic cytogenetics, even if there's a big network, but you still need to have the cells and culture, and you need days for that. So maybe not as accurate perhaps, but we thought we may be able to take advantage of the modification of gene expression in white blood cells to provide an estimate of those. So what we've been doing is to develop new protocol and use new technologies to develop rapid, high throughput, and keep in mind a cost-effective radiation dose assessment. So go directly into a gene called FDXR, which is, in my mind, I think by far the best gene for reporting a dose exposure. And a lot of work has been done ex vivo, but at some point you have to validate it, and that was a big work we've done to validate it in vivo using blood sample from patients. And we covered a large range of those, the largest we could. So going from TBI patient, so total body exposed, and then radiotherapy patient with different areas of exposure, going from high endometrium to much lower, like prostate or breast. And in much lower doses, we went to CT scan as well. So blood leukocytes, the white blood cells, were obtained from the blood, lysed, isolate the mRNA, and then we used a quantitative PCR assay, which is very sensitive to look at a specific gene to identify and quantify the RNA of this gene FDXR. That's the results we got. And I think it's fair to say that there's a remarkable sensitivity over a large range of doses here for this gene. You've got the CT scan here, and you've got the TBI here. So I think that was a good demonstration that at least these genes provide a very good dose estimate over a large range of doses. But obviously this is obtained while you have access to a control sample from the same individual before an exposure. Importantly, we had to calculate the dose to the blood, and that was done. And the dose to the blood here, we need to validate it in a way. So that's an experiment which probably has not been done very often. It was done with taking a blood sample from the patient before the first dose, the first fraction of radiotherapy, and we exposed that to a 100 milligram dose, because we thought that was more or less a dose we could expect from this endometrial patient. And that's here the result for a very limited number of patients, but that's not easy to organize. That's 3 patients here. And then this blood was exposed ex vivo. It was exposed obviously in vivo, and these are the dose the level of expression corresponding to the dose which we obtain. I think it's fair to say it's quite close. So we were quite satisfied with the good correspondence between in vitro ex vivo expression and the calculated dose and the in vivo measurement. Now, the part I'm going to conclude with is I was speaking about new technologies, and we're using this so-called third generation sequencing, where you can use your DNA or RNA molecules, and these molecules can be moved through a transmembrane protein called prorin here, and they make this sort of nanometer scale holes. And while these go through this hole here, it's going to modify a an electrical current. So I don't know what that moved. And what happened is you can then record these here and analyze them. And this real-time sequencing will allow you then to, if it's obviously analyzed on a computer, have a live sequencing data analysis. This is possible. So that's what we've done. We actually used a blood sample from 9 healthy donors, analyzed control and analyzed the 2 gray exposed samples. And this is a signature which we found. So we're obviously quite happy to have FDXR here. You can see as one of the top genes, as you would expect. Most of the genes here are upregulated, very few downregulated. But we have a total of 46 genes were found. So we went one step further, which is to look at not only FDXR genes, and the mRNA, but the variants. So the variants come during the splicing, which is, you can see here, which is a dynamic process where newly made precursor RNA are transformed into a mature mRNA, and the introns are removed here. But then you've got different variants. And it's very difficult to look at them by qPCR, but obviously with this long read third sequencing generation, it is possible. And we identify quite a lot of them. And the interesting one here are those with a green arrow. These two, they are less expressed than others, as you can see. But what is extremely interesting is that over between 60 to 100 million read of RNA in these cells, we couldn't detect any in the controls. So these transcript variants are upregulated in a dose-dependent manner. We confirmed their expression in vivo in radiotherapy patients. And they, as I said, have no detectable endogenous expression. And this is really really good, because one of the limitations we have is the sort of background noise of expression in control, but these don't seem to have any. So obviously that will have to be confirmed, but there's more to come, I think, from these. And last, based on what I just described, I think we are relatively optimistic we can develop this biodosimetry sequencing platform. And the idea is to develop a portable and high throughput real-time analysis, and again, cost-effective tool. So you could get this blood sample on site if need be, and then RNA processed, sequencing being done, and you can see the size of the one of the smallest sequencer we can use. So definitely portable. And then you do the obviously detection of electric signal, the alignment to identify the RNA, and then here what you have, and I take quickly an example, you can read in real time over a period of time, perhaps minutes, perhaps more. We'll have to confirm that in the next years. But what you can see here for individual number 1, over this period of time, the number of count, the sequencing of this particular mRNA goes above a threshold, and that means has been exposed to a high dose and need a medical attention. Individual 2, a lower dose, because it takes more time to get to the same number of reads. Non-exposed would be the green one, and perhaps the last thing which is really exciting is you actually accumulate a lot of data. Every single RNA is there, so you can also take advantage of that. Even in those individuals not exposed, you could pick up an immune signature of a biological warfare agent, for example, viral or bacterial. So here you would have that. So it's quite interesting for triage purposes. And I will stop there. Well, this is what we call it tachyon for now, and you probably speak to the physicist. And thank you very much. And I don't have time. I have a very long list of colleagues in the group, department, and externally I would like to thank. That's it.
Thank you so much, Christophe. Thanks for everyone who presented today. I think This really showed how much the large scope of work that you all do, and I think there's a lot more to explore. So we're at 10:10. What I propose we do, a lot of people have not signed off yet, which to me indicates that there's some interest in discussion. I suggest that we go till 10:15, so we have 5 minutes for discussion, and then the early career and trainee group can split off at that time. Are the speakers available for that? Is there interest in that? Okay, great. So in that case, why don't we go to some questions for Christophe? I think there was a question from Stephen Barnard. How long does the gene expression signal last following low dose /CT exposures?
Yeah, well, obviously, this is not a long-lasting signal, and in vivo, it has not been possible to assess it in human, but ex vivo, when you can't keep samples for very long, I can certainly say 3 days is fine. And obviously, if you lower the dose, then the signal will fade away. And if you go for very low dose, around 10 milligrams, then obviously things will disappear quickly, or we will lose the level of detection. So we're speaking days, and there's probably more to come from that. From these, splicing variant might be different, but yeah, I can't say more from on that for now.
And I see in the chat, Liz has posted more information about the chromosome dosimetry service and related research. And then just one more question from Mark Liddell, and then I'll open it up to a broader discussion. Mark wants to know, Christophe, how sensitive is RNA splicing to assay at low dose?
Okay, so obviously these are rare transcripts, so you need to sequence more to detect them. But at the same time, because there's no background, I think it has a big advantage. You don't need to count many. Maybe you need to count 1, 2. As soon as you see one, you know something happened, and then has been an exposure. That's what I can say at this stage. And obviously, there might be confounding factors and other things going on. But I think, yeah, the following years we will say, and I think it's really nice to see these cells and in vivo as well. You've got this this really radiation specific in that case response which you can detect.
Thanks. All right. Unless anyone has anything they want to say out loud, I've got another question in the chat going back to external dosimetry, I believe. So Rob Hayes wants to know, so I think this is a question for you, Jonathan. What are the dominant confounding factors in external dose reconstruction associated with limiting, reducing detection limits, and what are the current decision limits today? And I know Jonathan's got a lot of questions in the chat. He's also simultaneously trying to answer.
Sorry, I'm trying to do, sorry, can you ask it again? So I'm trying to do 2 things at once.
Yeah, sorry about that. Yeah, so I think just the last question, what are the dominant confounding factors in external dose reconstruction? Sorry to catch you off guard there.
Perhaps whether I'm understanding the questions correctly. I mean, the current decision limits within external dosimetry tend more to be in terms of operational limits. So they're set of the maximum dose that an individual can get per year. Now, the answer to that question depends to some extent on who the individual is. So a member of the public, for example, versus a classified worker. For example, the classified worker can receive 20 millisieverts a year. A member of the public can receive 1 millisieverts a year from essentially the same exposure condition. So I'd say that they're the closest I think there is in dosimetry to a decision limit. If the question is more of the decision limit for a particular instrument or dosimeter itself, then again, of course, that depends very much on the sensitivity of that instrument or dosimeter. Perhaps that's not answering the question, but maybe Rob could come back and let me know whether I've answered that or not.
Okay. Yeah, we'll see if there's more discussion in the chat on that question. And I see Elizabeth, you raised your hand.
Yeah, thanks. Well, thanks to everybody. This is really, really interesting. But yeah, I had a question for John. I mean, you mentioned in your talk that these new quantities will have an impact on the analysis of past epidemiological studies, and I just don't see it, because the past epidemiological studies were based on what was measured at that time, and then all these assumptions about how you go from HP10 to those two specific organs based on energy, geometry, etc. You're not going to have the new quantities for the past. So how do you see this affecting the analysis of past studies?
I suppose the answer, okay, it's a very broad statement to make that it will affect epidemiologic analysis, because clearly it will depend an awful lot on exactly what those analytical and what those analyses were. You know, of course, I think the suggestion of the analysis I was thinking of is if an individual, for example, would be to make analyses of how occupational doses were to change over time, so how to change over the last 20 years and over the next 20 years. Then, because the dose quantity has changed, it's quite possible that, as I said, even if the same individual has done exactly the same role for the last 20 years and does it for the next 20 years, if the dose quantity has changed, potentially the dose they received has changed, and therefore the dose that gets put on the dose record for that individual has changed. So it's only, I think, epidemiological analysis over time that you could start getting this mismatch of data, and you need to specify exactly which type of sievert, as it were, you were referring to. Are you referring to HP10 or HP, for example? But yes, I mean, clearly there's a very wide zone to say, of course, not affect all epidemiological studies. Certainly one that was completed from 20 years ago, of course, won't be changed by making a change now.
And are you expecting very big changes between HP10 and these new quantities? I mean, is that going to really affect radiation protection, or are we just doing a little bit better?
Again, I think that's a very hard question to ask, because it's still very context dependent. So for some fields, the change in the old dose quantity to new dose quantity, or the current to the proposed, will make no difference, because the values of the conversion coefficients essentially are energy dependent, angle dependent, radiation dependent, and so on. So for some fields, it will make no difference. In others, it could be almost an order of magnitude difference. So it's possible perhaps to contrive exposure scenarios where there would be very, very large differences. I mean, this of course was the motivation of ICRU in developing these new quantities, that the idea was that in some circumstances the current dose quantities do greatly overestimate or greatly underestimate the effective dose, which is of course the quantity that really we're interested in monitoring for people. So there was the attempt to smooth out those large differences in these new quantities.
Thanks. Thanks for all the discussion. We're over 10:15 now. So in respecting the session for trainees and early career researchers, I'm going to close out this discussion. I think there's a lot more to discuss, and I think there's some things in the chat that haven't been addressed yet. So I'll see if I can coordinate with Rania about getting some answers out to those questions at a later date, if that's okay with everyone. And I see more questions and discussion coming in, which is great. But yeah, I think I'm going to cut it here. Thank you everyone for participating. Thank you to ISoRED for organizing this. Thanks to Rania for organizing this. And we hope to see you all at our next sessions that start in April.